Hi everyone, in this video I want to talk about the details of nucleic acid structure. You're probably all familiar with the base pairing rules that A always pairs with T and G always pairs with C in terms of the nucleic acid bases. I've put two little lines here between the AT and three lines between the GC base pairs because, as you'll see when I draw these out, the AT base pair has 2 hydrogen bonds, whereas the GC base pair actually has 3. So here I've drawn the purine structure of the base A, and what I want to do is I want to draw out how it hydrogen bonds to T. So in drawing the structure, you have to rotate the pyrimidine about 30 degrees. So here, if I have A, I've rotated this about 30 degrees this way. And what that does, it positions a lone pair of electrons on this nitrogen of the A to the NH of T. So this is the nucleic acid base thymidine, or thymine. And it also positions the carbonyl of T next to the exocyclic amino group of A. So in the AT base pair, there are 2 hydrogen bonds. Okay. Now, if we go over to the G structure, this one actually has 3 hydrogen bonds when G is paired with C. So let me draw that out. Again, we're going to rotate our pyrimidine structure a little bit. And need to add a couple atoms here. And what this now does, it positions the carbonyl of G next to the exocyclic amine of C. This is C, by the way. The nitrogen here in the ring is next to the NH bond, and then likewise we have the exocyclic amine of G next to the carbonyl. So this has 3 H bonds. Okay, so just remember that base pairing rule. A always pairs with T, G pairs with C, and this is actually important, the fact that AT base pairs have only 2 hydrogen bonds, GC have 3. Now, where the squiggly lines are attached to each of the nitrogens, recall from a previous lecture, that's where these bases would attach to the sugar, in this case the deoxyribose sugar if we're talking about DNA. And then in the next part of the video, I'm going to show how these now can form a linear structure where you have multiple nucleotides linked together. So if we were to draw out now the sort of secondary structure of nucleic acids, which is going to be the polymeric sort of linear shape of many nucleotides linked together, let's just start up here with a phosphate. And what I'll do is I'll put on a deoxyribose sugar. So I'm going to make this little figure a part of a DNA molecule. Okay, so this would be called the 5' end up here. Notice we have a 5' phosphate. It's attached to the 5' hydroxyl, so that's 5', 4', 3', 2', and 1'. And then we're going to have our base attached here. Now, since I just, in the previous part of the video, I drew out all of the bases, I won't bother drawing that out. But let's say we have an A right here. Okay, that's one nucleotide. Now, if it's going to be connected to another one, this would be followed by the phosphate. So again, I would draw out another deoxyribonucleotide. And again, the 2' position would have an H here, like it does here because we're talking about DNA. And if I'm in DNA, I could have a T. Okay, next nucleotide could be drawn like this. So it's a repetitive polymer of multiple nucleotides. And I'll just draw 3 of these out. And let's make this base G. So again, we had the 5' end here. We're going down, and notice this now terminates in what's called the 3' end. If this, say, were the end of my little short piece of DNA. Now, when DNA wants to hydrogen bond and form the two base pairs between A and T up here, 2 to an A and 3 over to a C, what will happen is that the structure will now run in the opposite direction. Now, I won't draw out all of the sugars and phosphates, but I'll just sort of draw a line indicating the phosphate sugar backbone. And then I'll just draw something coming over here and write a C. Okay, across from the T will be an A, and across from this A would be a T. Okay, so there would be 2 hydrogen bonds between each of the AT base pairs, and then there would be 3 hydrogen bonds between the GC base pair. And again, I'm showing my strand going on this side in sort of a direction going downwards from 5' to 3'. This strand would be anti-parallel, so my 5' end would be here, and it would be running in the opposite direction. So when nucleotide strands join together to form these hydrogen bonded patterns, they do so in an anti-parallel fashion. And this is actually what Watson and Crick came up with in 1953 when they published their proposed structure of DNA, is they figured out that you had to switch the orientations of the strands, and they always found that A always paired with T and G always paired with C. So what Watson and Crick found in their structure, and this has been borne out in many, many experiments to verify the structure of DNA, is that if you have a strand, let's say that starts with a 5' end, what it will do is it will form an anti-parallel structure that's then called the alpha helix. And this is a very crude diagram. Your textbook would have a much nicer diagram here. But the strands are anti-parallel, so notice I've switched the 2 ends here. And in between, what's actually holding these together are those base pairs. Okay, and each strand is composed again of an alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. So what really is defining the sequence here then is the actual sequence of bases. So if we were to draw out a strand of DNA, we could just dispense, we don't have to draw all of the structure, we can just write the letters. So let's say we had a sequence that was written from 5' to 3' in the following way. A, T, T, G, C, C. Now by convention, we always write nucleotide structures from left to right, from 5' to 3'. And so if I wanted to now draw what the opposing complementary strand would be, that would be the following. Now again, it's going to be anti-parallel, so whenever there's an A, we would have a T across from it. This would be A, A. G's are going to pair with C's, C's will pair with G's, and so on. So this is the so-called complementary strand. And when DNA forms this double helical structure, again, the strands are anti-parallel. Okay, one little trick I'll just mention, if you were to see a nucleotide structure drawn in the following way, let's say A, U, G, G, U, C, C, whenever you see uracil or U in a sequence like this, you need to immediately recognize that we're not talking about DNA anymore. We're talking about RNA. So this is an RNA strand. Now RNA can form helical structures like this over short distances. They will often form hairpin loops, but usually RNAs are typically single-stranded. So when you see, again, this written with a U in it, the 5' end by convention is on the left, 3' end would be on the right. So that kind of wraps up the basics of the nucleic acid structures, the sort of linear secondary structure, and then this folded helical structure. Take a look at your textbook or take a look at it online just to look at the details of the alpha helix. It's really a beautiful structure. All right, so that kind of summarizes everything. Make sure you get some practice looking at these and being able to draw them. Good luck.